Okay, so we're now into the demo of looking at genes and transcripts in Ensemble. So I've just stayed on this region view that we were looking at before, because I just wanted to point out that if you are interested in finding out about a gene that you found through a region, or any other feature that you found in a region, you can just click on the feature, whatever it is, and it will get you a pop-up which will take you to more, it will give you more information, and in some cases there will be links, so we could find out more about this gene this way. In this case, we're just going to search for a gene, so I've put the gene we're looking for, ESPN, into the search box up here, and if I just hit return, it takes me to my search results. I get quite a lot of search results, and it puts the human gene at the top, followed by the human transcript, which is fine because we are, in this case, interested in the human gene. But if you were looking for another species, you might want to use this option to restrict by species, or if you're looking for something other than the gene, you might want to restrict the category. But in this case, I'm just going to click on the gene to go to the Gene tab. So Ensemble has a tabbing system. So every time you open a new type of feature, you get this tab appear in this blue bar here. So we've already got a location tab. We've now opened a Gene tab. If we were to then click on a transcript, we'll get a transcript tab. Click on a variant, get a variant tab. This is very useful if you're exploring multiple kinds of features. So if you're looking at a gene and then you look at the transcript and you want to flip back to the gene, you've got that very easy link there for you. So we have some information about the gene here. We have this transcript table which we can expand. So we can see a list of all the transcripts. We'll look at that in more detail later. So I'll hide that again. And we have all of the transcripts shown as a graphical display. We have on the left a menu of all the different displays, all the things that you can look at for our gene. And the first one I'm going to go to is sequence, which is here near the top. So the sequence it displays for us is the genomic sequence of the region of the gene. So it gives us a FASTA header telling us we're on a chromosome on GRCH38, chromosome 1, start, end. And this 1 here tells us we're on the forward strand. If we were on the negative strand, it would be a -1. Then we have 600 bases upstream of our gene of interest. And then we have the first exon of our gene of interest starting here. So all the exons are highlighted in this kind of peachy colour. And the exons of our gene of interest are shown in red. And you'll see that we've got a neighbouring gene has an exon, which is in this upstream 600 bases. If I scroll down, I get more exons of ESPN, but also, because biology is fun like that, an exon of a neighbouring gene too. I won't scroll all the way down. Just like the region view, we can configure this page. So if I click on the configure this page link here on the left, I can add things like variants. So I'm going to pick yes and show links. And I can add line numbering. I'm going to put relative to this sequence. And just as before, I click anywhere outside. And now it's going to load with my variants. It's giving me my line numbering. Generally takes a little bit of time to add the variants on. While I'm waiting for that to load, I'll just point out this download sequence button. If I click on this, I actually get two options of file format. So I can get FASTA or I can get rich text format. So the FASTA just gives me plain FASTA, and that's useful if I want to put it into some kind of sequence analysis tool. If I pick rich text format, it appears with all this highlighting on it. And so that's if I want to visually analyse it in something like Word. I don't want to download anything today, so I will just hop out of my menu by clicking outside of it. So now you can see it's loaded with all the variants highlighted across the sequence. There are a lot of genetic variants in human, and we will talk about these in the Exploring Genetic Variation talk, which is next week. So I mentioned Go terms. You can find the Go terms using these ontologies links over here. So if I go into Go biological process, so there's three categories of Go terms. There's biological process, which is what the protein does, cellular component, which is where it does it, and molecular function, which is kind of how it does it. So in biological process, we've got things like sensory perception of sound, locomotory behaviour. And we can also see what evidence was used to annotate it. So this one was inferred from direct assay, this one from mutant phenotype. And I could click on the transcript IDs. I could also put these terms into biomart or view them on the carrier type. So we can also see this gene in other databases. So if I click through onto this external references, I can see links out to other databases, which includes things like Expression Atlas, where you can get gene expression data across the body, MIM and MIM morbid for descriptions of the gene and things like that. So we can also, I mentioned Expression Atlas, we can actually see there is a display of this here if we go to gene expression. This is just a small widget taken from Gene Expression Atlas. If you are interested in detailed gene expression, I do recommend visiting Expression Atlas for this. And here we can see a number of different experiments, and we can see tissues along the top, and the colours highlight the level of expression. Also, you'll notice as I put my mouse over this is probably a better example here, you can see the tissue highlighted on the little human model there. So if you're not sure where a tissue is, this can help out. And if you're looking at sex-specific tissues, you can switch the gender of your little person over here. So the gene, as I mentioned, has quite a lot of transcripts. If I go into Transcript Comparison, I can compare these. So at the moment, nothing is picked, but this button here allows me to select transcripts, which I'm going to do. And I can add them one by one like this, or I could select, for example, all the protein coding transcripts. And so now I've got all the protein coding added to my list. If I save and close, what I get is a sequence view. Now I think this is really useful for stuff like designing RT-PCR primers. If you want to design primers which are specific only to one particular transcript of a gene, or that will cover all the transcripts of a gene, this can be a way to do it, because this shows you in sequence form which bits of the gene actually turn up in each of the different transcripts. So here we've got the beginning of the gene, we've got the UTRs, and you'll see that 3 of the transcripts are starting, 1 right at the beginning and 2 very near the beginning. But then all of them start being translated at the same point. So each line is 1 transcript. So we've got the gene sequence along the top. Each line is one of the transcripts. And obviously it's a very, very long sequence, so it goes over multiple lines. So now we're into the coding sequence for 3 of these. It does give us the variants as well. And as I go down the page, I can see introns shown in this grey. I can see regions where there's lots of transcripts as well. So if I go back up to the top, I'm going to open up the transcript table and we're going to look at what transcripts there actually are. So here we can see a list of all the transcripts. You can see that at the top we've got this ESPN 201, which is protein coding. We can see it's got a CCDS, it's got a UniProt protein linked to it, and it's got RefSeqs linked to it as well. So we could click on any of these links to explore it in other databases. We can see that it's a transcript support level 1. And if I hover over that, you'll see there's a description of what that means. It's part of GenCode Basic, and it's an APRIS P2. So this means that APRIS, I think it's probably one of the, probably the principalized form, but it might not be. They're not totally sure on that. But if you hover over them, you'll see what each of these mean. If I were to pick just one transcript to study from this particular gene, it would be ESPN 201, because it is coding, because it has this TSL1, because it's got this APRIS annotation. And so these are the criteria I would use to pick. It's also the longest. So then I would click on this identifier to go to the transcript tab. So now you'll see I've got this tab appearing, and I'm going to hide the transcript table because I don't need it anymore. And I have a different menu down the left-hand side with things that I might want to see for my transcript. One of the things I might want to see is how well supported it is, how likely we think this transcript is real, which I can go to in supporting evidence. So you can see the transcript model, and then we can see all the bits of data that we use to create it. So we have things like this NM, which is a refseq. If I click on it, you'll see that it tells me it's refseq, and it gives me a link so that I can go and see what it is. And there are some bits of evidence that we used to support the whole thing, and some bits of evidence like these ones over here, which just support fragments of it. And actually this is the exon supporting evidence from Havana, from the manual annotation. And you'll see a lot more kind of bits of fragment pasted together if you look at the evidence that's used by Havana. So we can look at sequence for a transcript as well. I'm going to first go to the exon sequence here. And this gives me a table showing me the introns and the exons. And the coloured highlighting shows me we've got UTR here, and this is the start of our translated sequence. And so you can see the exon numbering, the start and end, and the sequence of each one. Again, this is very nice for RT-PCR. The Cdna view, which is just underneath the exons view in the menu, is, I think, really nice for looking at variation because you can see the context a bit better. So in this case, we have not just, we have 3 lines. The first one is the Cdna, and it's better if we look at a lower down line. The second line is the coding sequence, and the third line is the amino acid sequence. So by being able to see the codons, which are highlighted, you'll see in these shades of yellow, you can then kind of compare the codons that you'll see and the variants that you might find within them to the amino acids that they affect. So I think it's really nice for looking at variation data. You'll also see just here we have a change in our exons. So we have a switch from the black text, or sort of greyish text, to the blue text, which tells us that we're switching exons, and it switches back and forth between the black and the blue. So we can see the transcript in other databases just like we could with the gene, and we go to general identifiers for this. And this gives us things that are linked to the transcript but also to the protein. So we have things like Human Protein Atlas, UniProt and things. If there was a structure available in PDB, you'd have a link to the PDB where you could go and view the protein structure. We do have some information about the protein. So if you go into protein summary, you can see the domains that are found in the protein when that decides to load. So we have, again, alternating shades give us the exons. So this is our protein model. And we use Interpro to predict the position of all the different protein domains. There are loads and loads of different algorithms that form part of Interpro, which have different methods of predicting protein domains. Some of them are really good at one kind of protein domain and less good at others. And it really depends on what protein domain you're interested in as to which is the most accurate. What you generally find is that you'll get an approximate region, such as over here where we have this ankyrin repeat domain, where all of the different methods of predicting protein domains have identified it, but they've given it different boundaries because they're working ever so slightly differently. But I know for certain that there is an ankyrin repeat domain somewhere over here. If I wanted to kind of decide exactly the coordinates of it, I would probably try and investigate more about Interpro and the different these different algorithms, so SMART, PRINCE, PFAL, and which one best serves my purpose. If you don't really like graphics, if you prefer tables, you can get exactly the same data by going to domains and features, but it gives you a table with start and end coordinates. That's everything I wanted to show you in the gene and transcript tabs. You should have another exercise to look at working on genes and transcripts, and then when you've done that, we'll have a look at Biomart.